Okay, hello everybody. I'd just like to present a short video on some of the general principles of cell signaling. This is just by way of qualifying this. It's not a replacement for reading the textbook, and it's not a replacement for coming to the lectures, but it's just something I thought that might be useful to you. So effectively then, let me just describe to you some of the generic principles of cell signaling. Um, what we mean is that if we consider a cell, the signal is typically outside of the cell and comes to the cell. So the cell has to detect the external cell signal. So there's a specific class of molecules that can pass through the membrane, which we'll talk about later. But in terms of generic cell signaling, we tend to think that the signaling molecule is detected by these protein receptors, which are embedded in the plasma membrane of the cell. So the external signal molecule binds to a protein receptor in the plasma membrane of the cell. Now, that extracellular signaling molecule doesn't pass through into the cell, okay? So it binds to the protein receptor. Now, typically at the protein receptor, there's a change in conformation of some description may occur within the protein receptor, and it's this change in conformation which not only changes on the outside of the cell, but it changes on the inside of the cell, and that's how you get the internalization of the signal. And typically there are some other factors which are also embedded in the cell membrane, which that newly that new structure of the protein can interact with, and then that is typically some membrane events. And effectively what happens is we activate a series of these kinds of interactions between proteins, and typically these proteins are not all membrane bound, but some of them are cytosolic, so that the signal is transduced into the cell, into the cytosol of the cell. Now, the end point of the signaling can be in the nucleus or in the cytosol. Typically, the signal reaches this endpoint, and we have some event triggered, and we have some change in cell phenotype or some biochemical phenotype that we can observe using some technique. And then typically that we have to have an off switch, we have to have the signal being terminated at some point as well. So for virtually all signaling pathways that we will discuss, we will begin looking at the detection of that signal and work through that particular signaling pathway to its target and the termination of that signal. So that's the sort of a generic thing we look for in all of the signaling pathways that we consider. Here's a very simple schematic to represent what we've just been talking about, and this is taken from the Hancock cell signaling textbook. So here we have an external signal of some description, which may be a photon of light coming into an eye cell. It may be a hormone binding to a receptor, or it may be one of many other small molecules or proteins that can interact with receptors. Here we have the receptor. The binding of the signal to the receptor causes a conformational change within the receptor. It may activate other things on the membrane, but effectively the signal is transduced internally and then will lead to a series of events, a cascade of events, which may end in change of gene expression, or it may end in the change of a protein activity within the cytosol or cytoplasm, and typically that could be enough to cause the phenotype. If we do get gene expression, then typically we have new proteins produced, which also cause a change in the cell, leading to a change in phenotype. So effectively, once the signal is internalized, we can sort of say there are two broad categories of responses, one which ends in the nucleus, resulting in new gene expression, new protein expression, and the other which goes through to the cytosol, which typically will cause a post-translational modification of an existing protein, such as protein phosphorylation or something like that. Like I said, I've been encouraging you to read appropriate chapters from other textbooks. One of the really good textbooks for cell signaling is Molecular Biology of the Cell by Alberts as the lead author. This is a figure from Alberts which refers to the same content that we've been discussing in Hancock, our recommended textbook. And here we have a signaling molecule binding to a receptor, the internalization, the change in protein structure referred to by these little pink lines here. Something's happened here, and then that allows either a cascade of events leading to DNA expression, RNA synthesis, protein expression, or it leads to a series of events which alters protein structure or protein function of an existing protein. And either of these outcomes is a change in cell behavior and a change in cell phenotype. The thing I like about this diagram is that it emphasizes that the cytosolic events are quick to occur, whereas new gene expression and protein synthesis is a slower process, so we have different time frames for these different responses from the signal. And in some cases it may be that one signal leads to both of these occurrences, or it may be that the signal leads to a single occurrence. So here we have a slide which is the still talking about general principles of cell signaling, and what we're saying here is okay, so if you consider a cell in your body, it's exposed to many different signals, all the time. There could be lots of signals happening in the body, but that cell is only responsive to a subset of signals. There's only certain signals that can be perceived by that particular cell type. Okay, so an important principle of cell signaling is that the cell is specifically targeted by the signal. Okay, or not specifically targeted. It's particularly receptive to a single signal or a single collection of signals. Okay, typically a single signal, such as a hormone molecule, may lead to more than one response. So it may activate different cell types, or it may activate different pathways within a single cell. Now, when we start to look at cell pathways in a bit more detail, what you'll start to see is some of the players of one signaling pathway also are players in other signaling pathways. So we have the concept that different signaling pathways may talk to each other through these common partners. So if there's a particular protein involved in pathway 1, that same protein may be involved in another pathway, and therefore you can jump from one pathway to another by that common member. And also another important concept in cell signaling is that we don't have a single signaling molecule activating a single protein within the cell. Typically we have an amplification of that signal, so that a single signaling molecule may activate many copies of a protein, and we'll look at that in the next couple of slides. So what kind of signals are cells perceiving? Like I said, they can perceive, signals are very diverse, so cells can perceive, some cells can perceive light, some cells can perceive small molecules, and some cells can perceive fairly large protein molecules, such as hormones and things like that. And so here I've got must be specific to relay a defined signal, only recognized by the target molecule machinery. That's a fairly convoluted dot point there. I guess what I'm trying to say there is that a good signal is one that targets the target cell and doesn't sort of cause other cells to respond. So that it's a specific response, so that there's only one or two types of receptor molecules that respond to the particular signal, so that we don't activate the wrong cell to respond, is basically what we're trying to do here, what I'm trying to say. Okay, so we often talk about what makes a good signal. It's important to think that for all of these signaling pathways, it's okay to activate the signaling pathway, but we also have to consider how that signaling pathway is going to be switched off. And I've put some big clunky switches here, such as that your emergency power button. I mean, how do you turn things off quickly? So we'll look at some methods the cells have to turn off signaling. So the examples that are quickly listed here are, one of the second messages that we're about to talk about is cyclic AMP, and the cell has a mechanism to break down the cyclic AMP and therefore turn off the signaling. We'll talk about the release of calcium from cells or within a cell to activate a signal, and we'll also talk about how the calcium can be sequestered by the cell and sort of made unavailable so that you can turn calcium related signaling off. So we've got to consider how signaling is turned off as well. Okay, so when we talk about cell signaling, when we talk about the signal binding to the receptor, there's a terminology that then says the second messenger in the signaling pathway is whatever it might be. Okay, so typically cyclic AMP was referred to as a second messenger. So there was a range of different signaling molecules that could bind to particular cells, and often one of the first things researchers identified as occurring was the activation of or the cells being able to make this molecule cyclic AMP. So this CAMP here is cyclic AMP. Now you've come across ATP before in biochemistry. ATP is the energy currency of a cell, and here we have the structure of the ATP. Now the TP stands for triphosphate, so it's adenosine triphosphate. Here's your 1, 2, 3 phosphate groups attached to the adenosine base and the sugar here. So here's your adenosine triphosphate. Now, for the purpose of signaling, what the cell does, it cleaves the triphosphate to a monophosphate, but we also have this cyclase activity, adenyl cyclase, so that the phosphate is internally attached there to the is that the 3' of the sugar here. So you get this cyclic compound here where the phosphate group is internally bound to the same sugar. Okay, and that molecule is your cyclic AMP molecule, and that has an important role not in energy transfer, not in sort of energy metabolism, but in cell signaling. Okay, so this is your second messenger, and that's how you activate that second messenger. But we also have in the cell a phosphodiesterase, your cyclic AMP phosphodiesterase. Now, and the phosphodiesterase cleaves this ester bond here to give you plain old AMP. So this is cyclic AMP, and this is AMP. So whilst the molecule is in this form, the cyclic AMP is active in signaling. And I apologize if I'm waving the mouse around like a crazy person, but there you have it. So effectively, this is the turning on of the signal here. The activation of the adenyl cyclase is part of the signaling pathway, and turning off the signaling pathway, part of that process is the activation of cyclic AMP phosphodiesterase. So here's an image from, again, the Hancock textbook, which discusses the initial signal giving rise to a second messenger here. And an example of the second messenger I've just been discussing with you is the cell manufacturing this cyclic AMP from ATP. So here we have an enzyme, which will be your adenyl cyclase. So we have the signal binding to a receptor, the receptor activating an enzyme, and this enzyme is then converting ATP, which isn't shown here, into something such as cyclic AMP. And then that goes on to activate or do something else in the cell. Another mechanism of releasing a second messenger is not to manufacture that second messenger chemically, but to release that second messenger from a store within the cell. So in this instance, the second messenger isn't cyclic AMP, it's calcium, just calcium ions. So here we have these little vesicles in the cell which store calcium ions so that the calcium ion is not available. But as part of the cell signaling pathway, we have the signal binding to a receptor activating something which then causes these sequestered calcium ions to be released. And that can be released externally, as shown here, or internally within the cell. And depending on the pathway, that will then give you a different kind of response. So there are two examples given here of second messengers. The first is the cyclic AMP, and the second example is the calcium ion. So typically we tend to refer to the signaling molecule as the first messenger, and then a bunch of things happen which is now better understood than it was when these terms were coined, and then part of the process is the manufacturing of that second signal, or the release of that second messenger. So I guess now we're going to have a quick discussion of what is the relationship between these cells that are talking to each other, whether they're close in space or distant in the body, and how that signal is passed on from one cell to another. Now there are two broad categories of distance, or if you like, we can discuss here. The cells can be not touching each other. They may be distant or close, but they're not in physical contact with each other. Or the cells may be in physical contact with each other, and the signaling mechanism is reliant on the fact that they're close in space. So let's quickly look at these two categories. So for cells which may be distant, we can have endocrine, paracrine, or autocrine signaling. Now you may be familiar with these terms from biodiversity or some other subjects you may have done. So in this case, we're talking about a signaling molecule releasing something which travels a distance to reach the target cell. And typically it'll travel a distance to reach other cells, but those cells won't respond. We talked about this idea of specificity from that convoluted dot point I had earlier. So we'll have a look at this idea of the target cell and the proximity of the target cell to the main cell that's signaling to it. So when we talk about endocrine signaling, I'm sure you guys can remember endocrine glands and hormone release going through the bloodstream, and that's exactly what we're talking about here. We have a cell in an endocrine gland somewhere in the body that releases a hormone into the bloodstream. It travels a large distance. It comes into contact probably with lots of cells, but there's a particular cell in the body that's got a particular receptor, and that is the target for the signal. So in that case, we then have a range of target cells that can perceive the signal. I guess a couple of dot points here. The signal is transported by a circulatory system, a long distance to the body. That molecule is in low concentration. It's highly diluted, and typically we have growth hormone and adrenaline and other hormones which travel in this way through the body. There's another scheme of signaling referred to as paracrine signaling. Typically this is for cells that are close in space, and the signal will typically diffuse from one cell to another. So here we have a cell which is releasing a signal which is diffusing to some local cells. So typically these signals are diffusing within the immediate vicinity, and the sorts of molecules that tend to fit this category are growth factors, cytokinins, and some neurotransmitters. And again we have the third class of this thing, which is autocrine signaling, and this is typically the cell that releases the signal is the cell that perceives the signal. So effectively the signal is coming back to the same cell. And there's examples of how some cancerous cells may be expressing receptors for the signal it's releasing, and therefore is responding to its own signal. And that's referred to as autocrine signaling. Another scheme which is you're probably familiar with is this idea of signaling in nerve cells. So we have signals occurring here somewhere. We have an electrical impulse traveling along the axon of the cell, which is like an electrical signal, giving rise to a chemical signal which is released to an adjacent cell. Now, these cells are very close together in space, and often that signaling molecule has nowhere to go except to the receptors on the adjacent cell. So there's no dilution effects here. There's no long, even though we have traveling the distance of the axon, the signaling molecule itself has nowhere to go. It's just very direct signaling to the adjacent cell. So when we talk about direct cell-cell communication, there are two categories of this. In one instance we have these two cells which are talking to each other. This cell contains a signaling molecule embedded in its plasma membrane. This target cell has a receptor molecule embedded in its target membrane, its cell membrane, and these two molecules at some point align and bind to each other whilst those cells are adjacent, and therefore this cell is directly communicating to this cell. And again we get either a cytosolic or a nuclear response through this signaling pathway. And the last example I want to just quickly mention from the textbook is the signaling through these gap junctions. And again the book describes to you how there's a bunch of proteins which can form these pores in the membrane, and between these two cells these pores can align, opening up a channel between the two cells. Typically this channel is very narrow. It doesn't allow for the passage of large proteins, but allows for the passage of small molecules, such as our second messengers we've discussed earlier, cyclic AMP. I'm sure there are examples of this for the passage of calcium ions as well. And that's signaling through these gap junctions. Typically, if you look at the point where these gap junctions are, the membranes come together, and there's a series, there's many of these protein aggregates forming these channels, allowing the passage of ions between the two cells. And the last concept I'll just quickly mention to you before I wrap up is during the signaling pathway, we always tend to see this amplification of the signal, because when we talk about a cell, the phenotype of a cell is actually a very big change that's occurring to that cell based on a very small amount of signaling to that cell. So we have a small signal causing a large change. So it's almost obvious that there has to be an amplification of that signal to cause such a large change. So as we pass the signal from one component to another component during the signaling pathway, such as from a signal to the cyclic AMP, we have an amplification, so that one signal molecule leads to the formation of many thousands of cyclic AMP molecules. And then we'll discuss later how cyclic AMP can activate other proteins such as protein kinases, and typically will activate many protein kinases, which then go ahead and act on many substrates. So you get an amplification of the signal through this cascade of events. And again, here's a modified figure from the Hancock textbook. I've just sort of added a bunch of arrows to it. But what we're effectively showing here is that, for argument's sake, a single signaling molecule activates a receptor, which can then go away and can then move around the membrane and activate one or more enzymes, such as adenyl cyclase, and then each activated enzyme will then convert many ATP to cyclic AMP, so you get many thousands-fold amplification of this signal through the generation of many copies of cyclic AMP, which can then go ahead and activate many protein kinases, and protein kinases we'll then discuss later on will activate, and phosphorylate a bunch of molecules, producing a large phenotypic change within that cell. So not only do we think about the cell path, the signaling pathways being specific, within that pathway we also have amplification of that signal to activate many proteins within the cell. So typically we're talking about a signaling pathway, whereas a receptor activates an enzyme that causes a second messenger, which causes activation of another messenger, etc. And we look at this as a linear thing, but in reality we tend to have a single protein activating a messenger which may activate different things within the cell. Different proteins may respond to the same second messenger. So, for instance, cyclic AMP may activate more than one protein. It may activate protein A and protein B. And therefore we get a cascade of different pathways being activated from a receptor. And effectively, this is the idea of crosstalk can occur between signaling pathways. Typically, we'll study a pathway in relative isolation, but just at the back of your mind, be aware that there is crosstalk between signaling pathways. And you may pick up some crosstalk when you do your assignment. There may or may not be some crosstalk between the different pathways you're considering. And lastly, we're talking about signals passing through the cytoplasm of the cell. How do we think about the cytoplasm? Is it this, homogeneous liquid sort of thing, or is it a place where you can have concentration gradients that exist and that are maintained within the cell? Typically, it's not thought of as a homogeneous mix, but we tend to think, it's observed that you can have concentration gradients within the cell. So you can have, for instance, hotspots where when calcium is released within a cell, it doesn't diffuse across the cell in a homogeneous way, but it stays in a higher concentration in one part of the cell. So one way to sort of minimize crosstalk is having these concentrations of molecules within the cell that don't diffuse randomly around that cell. So you can have a pathway, where you've got more of some signaling molecules in one part of a cell. So this may reduce this idea of crosstalk, giving a bit more specificity to certain signaling pathways. Another way the cell maintains a signaling pathway is by having these proteins, which are typically referred to as scaffold proteins, which don't directly signal to each other, but they hold together molecules that do signal. Okay, and again in later lectures we'll talk about some of these proteins which hold together a series of what we'll discuss, these MAP kinase proteins. So these MAP kinase proteins are held together by these scaffold proteins, and these kinase proteins communicate to each other and not other proteins, because they are held close in space together by these scaffold proteins. So even though we have this notion of crosstalk between signaling pathways, the cell has mechanisms to reduce crosstalk, either through limited diffusion of reagents within a cell or by holding certain proteins together with these scaffold proteins. And that way you get these more specific responses to a signal. Okay, well I hope that's of some help to you, and I'll stop recording for now. Okay, thank you.